To enrich for circulating nucleated cells from blood, the microfluidic device is first primed with 1x PBS to rid of bubbles. Deionized water and 2% paraformaldehyde in 2x PBS are then run through the device via syringe pumps prior to loading of the blood sample. Blood is then processed through the microfluidic device and collected. Post-processing of the sample includes centrifugation and removal of supernatant. Finally, the sample is analysed via flow cytometry to produce FACS plots.
Hi, I'm William White from the Laboratory of Microscope Biotechnology in the Department of Bioengineering at the University of Lowell. Today we'll show you a procedure for the enrichment of circulating nucleated cells by lysis of whole blood via microfluidics. We use this procedure in our laboratory to study inflammation in various disease processes as to ensure enrichment of leukocytes and other circulating nucleated cells without loss or activation. So let's get started.
To begin this procedure, press fit access tubing of slightly larger diameter than the channel access holes in the inlet and outlet holes. Then, to provide the macro to micro interface, attach a 30 gauge syringe needle to the tubing on each of the inlets. Next, fill a 1 ml syringe with 1x PBS, making sure to get rid of bubbles. Connect the syringe to inlet 1 and push the syringe gently by hand until 1x PBS flows out of inlet 2. Then immediately clamp inlet 2 with an office binder clip. Continue pushing the syringe until 1x PBS solution reaches the outlet port of the microfluidic device. Once the solution flows out of the outlet, clamp the outlet tubing with another binder clip. Continue further pushing the 1 ml syringe until 1x PBS flows out of inlet 3, and clamp the inlet 3 port with a third office binder clip. The microfluidic device is now fully primed. Start calibrating the syringe pumps with the large Harvard syringe pump, setting the diameter configuration to 22.5 mm and the flow rate to 600 microliters per minute. For the small Harvard syringe pump, set the diameter configuration to 4.61 mm and the flow rate to 20 microliters per minute. Next, fill one 30 ml syringe with deionized water by withdrawing the solution directly from the 50 ml conical tube. Remember to label the syringe. Fill another 30 ml syringe with 2x PBS 2% paraformaldehyde solution. Again withdraw directly from a conical tube and label the syringe. Now connect the water-containing syringe to inlet 2 and the 2% paraformaldehyde in 2x PBS syringe to inlet 3, while making sure no bubbles are trapped in the tubing or in the microfluidic device. Then remove the clamps from inlet 2, inlet 3 and the outlet tubing. With all three syringes connected, proceed to put the two large syringes together on the large Harvard syringe pump. And set the 1 ml syringe on the small Harvard syringe pump. Place the outlet tubing in a 50 ml conical waste collection tube. Now turn on the large Harvard syringe pump holding the 30 ml syringes and let it run for 1 minute. Check that there are no bubbles and no leakage in the device or in the tubing. Then turn on the small Harvard syringe pump and let it run for an additional minute, here too checking for bubbles and leakage. Stop the pumps after 2 minutes as the microfluidic device is now ready to be used with blood samples. The blood samples to be analysed were collected as described in the accompanying written protocol and are used within 1 hour of collection. The first tube of collected blood is discarded to rid of false positive results. Remove the 1 ml syringe from the small Harvard syringe pump. Then, inject 1x PBS from the 1 ml syringe, filling the syringe needle to avoid introduction of bubbles. Fill a 1 ml syringe with 50 microliters of 1x PBS without trapping any bubbles. Then fill the syringe with 0.5 ml of blood. Keep the syringe vertical while filling to avoid mixing of the PBS with the blood. Next, connect the syringe containing the blood back to inlet 1. Wipe away any excess fluid and mount the syringe vertically on the small Harvard syringe pump. Be careful not to push the blood through the tubing into the device. Remove the outlet tubing from the waste tube and place it in a clean 50 ml centrifuge tube sitting in an ice bucket. Now start the run. Switch on the large Harvard syringe pump first and let it run for 1 minute. Then switch on the small Harvard syringe pump. The sample will start collecting from the outlet tubing into the 50 ml centrifuge tube. Let the blood sample in the 1 ml syringe travel completely through the device, which takes approximately 20 minutes. Then stop both pumps. Now that the run is complete, centrifuge the collected sample for 5 minutes at 350 times G at room temperature with the break off. Remove the supernatant by placing the pipette tip at the opposite side of the white pellet. Remove as much supernatant as possible, especially red cell debris, without disturbing the white pellet. Resuspend the sample in 1 ml of flow buffer containing 1% BSA and 2% paraformaldehyde in 1x PBS by pipetting up and down. Transfer the sample to a 1.5 ml Eppendorf tube. Microcentrifuge the collected sample for 5 minutes at 4 times G at room temperature. Remove the supernatant as carefully as in the previous centrifugation step. Finally, add 1 ml of flow buffer. Vortex to resuspend the sample and proceed to analyze the sample by flow cytometry. Now we'll show some representative results in the form of fax plots from a blood sample enriched by the microfluidic device with analysis using flow cytometry. The importance of enrichment by lysis can be seen by a fax plot where red blood cells cloud the scatter plot. Produced by the microfluidic enrichment protocol is a clean scatter plot with defined nucleated cell populations found in circulation. The red region represents lymphocytes, green represents monocytes, blue represents granulocytes, and the purple region has yet to be characterized. Failure to remove red cell debris in the protocol produces a cluster of events in the fax plot, seen in the cyan region. Populations of cells can be visualized in separate fax plots. Lymphocytes are stained with CD3 and CD4, monocytes are stained with CD14, and granulocytes are stained with CD66b.
We've just shown you how to set up and run a blood sample with the microfluidic blood lysis device. When doing this procedure, it's important to remember to run the blood sample immediately, to not allow the PBS to mix with the blood prior to entering the device, and to avoid introduction of bubbles into the channels of the device. So that's it. Thank you for watching and good luck with your experiments.